Good morning to you all. In the plant protection class, the last class we were discussing the rice diseases and its management. We are going to continue the part 2 of this section. So you all know there are several causes for the rice diseases. We can classify it into bacteria, to fungi, virus. These are three microorganisms causing the severe diseases, economically important rice diseases. Other than that, you can expect other microorganisms like nematode, root knot nematode, and white tip nematode. These are nematodes are also causing some economic damages in certain part of the rice cultivation areas. So today's class we are going to continue with the third economically important disease is the narrow brown spot, right? So this narrow brown spot is highly confused with the brown spot disease of the rice, but it is not much severe because if you are going to take any management aspect to control or manage this brown spot disease, easily you can manage these narrow brown spot disease, but the causal organisms are different. So the brown spot is caused by the Helminthosporium oryzae or Cotylobullus myabinus, and the causal organism for this narrow brown spot is a Circus fora oryzae. The Circus fora is an folifagus fungi. So other than this rice, you can expect there are different Circus fora species in different crops. So characteristic symptoms of this narrow brown spot is, so you can expect brown from the title of this is narrow brown spot means a brown spot. Normally brown spot lesions you can expect mainly in leaves. Occasionally or in rare cases you can expect in sheath, pedicel and glooms too. Right? These narrow brown spot characteristically, so brown lesion and they are short and linear. So this is an identification for from the picture you can easily identify, right? Not like a brown spot. Brown spot all over the leaf area you can expect a lot of spots, but here so characteristics small linear spots. That is an identification, but sometimes it is confused with an advanced stages of the brown spot. I mean the sporia. Therefore, if you are taking management aspects for the both diseases, so the management practices are common for both diseases. So the management options is normally farmers they are using these foliar fungicide, spraying of foliar fungicide, carbon diazem is in broad spectrum. So by using this carbon diazem or mancozeb in the rate of 2 kilogram per hectare, you can easily manage the brown spot, narrow brown spot, even blast disease, right? By using the single fungicide can control because this is a broad spectrum. So next disease is a rice seed blight. So rice seed blight is caused by the Rhizoctonia solani, right? Rhizoctonia solani is a folifagus fungi. You can expect seedling stages, especially the damping of his and seedling rot is caused by Rhizoctonia solani. This Rhizoctonia solani causes a seed blight, right, in the rice plants. So sexual stages of this fungi, we call it as an Thanatophorus cucumaris, right? Thanatophorus cucumaris. So this disease is economically important if your rice is cultivated in the wetland area or unexpected rain with a cold climate observed, right? In while cultivating the rice, you can expect this sheath blight, especially the flooded areas. If it is flooded suddenly, definitely you can expect this rice sheath blight. The symptoms initially, because the name is sheath blight, so you can expect the first symptom in the sheath, right? Where the leaves attached with the stem, you can be called as a leaf sheath. In this area, you can easily expect the first symptom. Initial symptoms are small. It's a ellipsoidal. It's an ellipsoidal means like long circle, right? Ellipsoidal or ovoid, close to round to ellipt, right? Round to ellipsoidal. So initial symptom is a greenish gray, right? Initial symptom is a greenish gray and water salt. It's like an oil patches, right? Like an oil patches is where you can expect mainly is where the water level is there. Why? So because Rhizoctonia solani produces an asexual spores. We call it as an asexual, not a spore, not a true spore. It's an aggregation of mycelium. We call it as sclerotia, right? So this sclerotia is having the certain characteristic of floating behavior, right? When the water level is there, it can float. So if it is float, then it can go and adhere. If it is touched with the this leaf sheath surfaces, they can initiate the infections. That's what you can easily accept these diseases in the waterline of the lowland peripheries, lowland areas. You can expect the first symptoms. The older symptoms are the ellipsoidal size will increase, right? Ellipsoidal size increase, then you cannot expect that greenish or water shock mark. Here, the grayish white center and light brown, dark brown margins, light brown to dark brown margins. So that means that the disease advanced in the older stages. So you cannot expect these watery marks. You can expect a greenish white center with the brownish to dark brown margins along the stem. Right? This is an advanced stage symptoms. So this symptom, if it is advancing from sheath to leaves, definitely you can, under favorable conditions, you can expect these symptoms even in the leaves. If you are observing these symptoms in the leaves, definitely you can find the lesions may coalesce and bigger lesions. That's what you cannot tell the definite shape of this symptom. So irregular outline with the depth of the whole leaves, mean leaves will become and drying and lying. So in the advanced stages, it may confuse with whether it is brown spot or if it is with the blast. So in the initial identification or initial observation only will give you the idea. But if the disease is advancing, you can expect the symptoms like, right, severely infected plant produce poorly filled or empty grains, right? So this is also the confusing symptoms with the blast, right? If it is the neck blast observed, you can find that poorly filled or empty grains, or even jealous temporal attack also will give the white head like the similar symptoms. Therefore, so this is in advanced cases, severely infected plant produce poorly filled and empty grains, right? This is a third. There are three symptoms. First symptom is in the sheath regions, you can expect ellipsoidal or ovoid shape with a water soaked and gray, greenish gray. Next, older one, you can find a grayish white center with a light brown to dark brown margin. So if the disease advancing, lesion can go, you can observe in the leaves too. If it is observing the leaves means, so the coalesce of these leaf sheath symptoms and irregular outlines with the depth of the whole leaves. Other than that, you can expect that severely infected plants also producing poor filled grain or the empty grain. So these are four symptoms are important. Other than that, one of the major important symptom is there. This symptom is present. You can definitely say this is because of Rhizoctonia solani, seed blight. This means formation of the sclerosis. So where you are observing the symptoms in the advanced stages, you can find that rusty patches like grayish color, grayish to white color, formation of the sclerosia. Sclerosia is a round like a mustard seed. In the advanced stages, it will become a brownish color like a mustard seed. Exactly mustard seed. So this is like sclerosia mean the aggregation of the mycelium, right? Aggregation of the mycelium. So the sclerosia is loosely attached with the symptom or mycelia. So in the symptoms means symptoms you can observe mycelia. No, in the mycelia, sclerotia attached loosely. Mean if you touch it or if the wind is shaking the plants, the sclerotia will fall off and easily it can spread with the water. So this sclerotia is having a characteristic specific character means it can able to float. If it is floating mean when the water is moving it can spread to adjacent field too, or if the adjacent field is got infected definitely you can expect your field will get infections, right? But for the initiation of the disease the rice field should have the water, otherwise you cannot expect this sheath blight diseases. So this is an easily you can identify or easily differentiate in the sheath, the blackish brown margins, so near the water lines, and the leaves, the irregular lesions, and it is get coalescent will get dry off. And the gray center, it is an identification or typical characteristic, like not like the brown spot. In the brown spot also, narrow brown spot also, you cannot expect this gray center. Only the gray center you can expect in another disease is fibricularia or rice blast disease, but the shape of this lesion is spindle shape in rice blast. But in sheath blight, here the irregular shape, but the gray patch center you can expect. So this is the formation of the sclerotia, right? So younger sclerotia and mature sclerotia. Mature sclerotia will become a brown like a mustard seed. So normally this disease is spreading through the water. So therefore, and soil movement is very less. But if it is sclerotia is formed while the disease advancing, definitely 10 to 20 years you can expect the disease, because so removal of sclerotia or the killing of sclerotia is very difficult. So that's what the initial observation and management of the disease is highly appreciated. Otherwise your field gets sick, and in forthcoming seasons also you can expect these diseases. So these first observations after plant reach maximum tolerance. So this disease where you can first symptom you will observe is not a seedling one. Where you can observe in when the tillering started. So maximum tillering stage is there, then definitely you can expect, because in between the tillers, sclerotia can go easily and is easily trapped. Right? If it is a single stem, there is no chances for the sclerotia to get in touch with the sheath region. So if it is the heavy tillering stage, definitely sclerotia can easily go inside and trap with the water. That's what these symptoms or disease infection is initiates when the tillering stage is advanced. So problem for the management bin, I already explained that one. This it forms a sclerosia. This is an aggregation of the mycelium. That's what this fungi is also called as a sterile fungi. Mean it will not produce the uh, earlier it was classified as a sterile fungi because it did not form any sexual stage. But later stages they have identified that they are forming the sexual spores. That's what it is called, it as a thanatophorus cucumaris. Right? Thanatophorus cucumaris is a sexual stage. So this it so the sclerotia can survive for a long period. I already told you it may be 10 to 20 years. Therefore, and also it has a characteristic of floating ability. If it is if it can able to float in the water, then it can easily spread. And also sclerotia was means dormant mycelium, no aggregation of the mycelium inside the hard coat. So if the sclerotia hard coat means, so it can able to survive even in the hostile environmental conditions. So and also this fungi, I already told you, this fungi is a polyphagous fungi. So therefore the removal is weed is important. If it is any plant or if the weed is there, definitely this fungal can survive even in the mycelia or as a sclerosia. Then the main crop is cultivating, it can easily initiate the infections. Therefore these are the things are very problematic managing of this disease. So if you are taking the pathogen into considerations, I told you there are two stages. So normally in the both stages, the fungal produces septate mycelium. You see here and there the septate mycelium, but hyaline. Hyaline means colorless in the initial mycelium, but later it will become a junk or it will become a yellowish brown, right? Yellowish brown when it is old. Earlier it is a hyaline, colorless, right? So it produces large number of spherical brown sclerotia. Earlier sclerotia is whitish, then it will become a brownish color. So confirmation of the disease. So if we are observing the symptom in the sheath, the first confirmation. The second one is if the symptom is present in the leaf, irregular shape with an gray center. Third is a formation of the sclerotia. These three are specific symptoms to confirm this disease, and also if it will not cause disease in the dry season. So if it is a wet season, then you can easily suspect this is because of this sclerot, right? Rhizoctonia solani is forming the, it causes a sheath blight in the rice. So conducive climates. So presence of the disease in. So I told you if the sclerotia formed, then it can survive in the soil. It may be 10 to 15 years. Therefore, if the soil, the disease inoculum is present in the soil, then it conduces the disease. And this sclerosia. This both. Third is a. So I told you this disease will not come in the dry season. So therefore, the relative humidity is high, 96 to 100, like a blast, right? So it favor the development of the sheath blight. And temperature, 28 to 32. So mean wet, hot season favors the development of the disease. And high level of nitrogen application, it means that you can expect more tillering. If it is more tillering, mean definitely your plant will become a sick, right? The third for next one is the present of irrigation water. This irrigation water is need, so because the sclerotia can able to float and can get in touch with the sheath and start the initiation of this disease. And growing of high yielding improved varieties. Normally the high yielding improved varieties are susceptible cultivars. If the susceptible varieties are present, definitely you can expect this disease. And late tillering or early internode elongation, right? So if the cultivar is an early internode elongation, mean easily you can expect more sheaths. And tillering, if it is a late tillering, mean and the advanced stages, if if it is producing more tiller, definitely you can expect this sheath disease. So these are the conditions favorable for the diseases. Or if you're taking the disease cycles, normally, so primary inoculum, sclerotia will be the primary inoculum. If the irrigation water is there or the rain water is there, or if if your field is get flooded, right? So in the very humid condition or dry in the daytime is 28 to 30 hot season. So mean humid and hot season favors the presence of the water favors the formation of mycelia or germination of this sclerotia. So once the sclerotia germinated, you will know there's normal infection stages, means it will produce germ tube produce prosoria and produces some toxic substances and enzymes and form the penetration peg, and it will penetrate and go in, and it will go into and advance into the plant cell to the intracellular mycelium. So then fungi will grow inside and multiply, and it will reproduce again and formation of the sclerotia. In this way the disease cycle is progressive. So sheath blight, why we are calling it as sheath blight is important, mean so because the sheath blight attack is there, definitely you can expect if it is an advanced disease attack, right? It's in advanced stages, unfilled, more unfilled grain, and a seed blight get infected, your plant can able the plant cannot able to withstand because it can able it will fall down. It means lodging, right? Lodging and the death of tiller. So if this happening, then definitely you can expect that the yield loss, so much of yield losses, poor milling quality, and low panicle length. Because if it is in the boating stage, if it is sheath blight is expected, definitely you can tell your panicle will not come out fully, right? Come out fully. So the reported yield, if it is a booting stage, if this disease incident is happened in the booting stage, 50% yield loss you can expect. So in the flag leaf initiation, it is if disease develops, definitely 20 to 25%. So flood with the high nitrogen, it can cause 20 to 42% yield losses if the disease. Therefore, you have to be very careful from the tillering stage and careful monitoring and proper amount of nitrogen fertilizer. Even easily you can minimize the disease incidence. Easily minimize the incidences. So this is, it has been reported that in Japan, yes, it is causing 20% in losses. In US, it is 50%, right? 50%. But this attack is less in Asia. So nearly in Asia, it causing nearly 6%. 6% yield losses. So because in the conditions are not favorable in Asian regions, tropical Asia, I mean, so the RH will not go more than 90, right? So therefore, this disease incidence is very high, very low in tropical areas. So these are the alternate hosts for this disease. So any crop, previously cultivated crops, is there got infected, definitely your paddy field, the more chances to get infections. Therefore, the folifagus fungus, this is a folifagus fungus. Therefore, you have to be very careful on remove cultivate when cultivating. So your field should be monitored from the initial point of cultivation to minimize these incidents further and advancement of the disease. So therefore you have to be careful on, so in which variety of the rices you are cultivating, right? And how much of seed rate is used, whether nitrogen rate is very high or not, or leaf symptom pattern. And if you're observing all these problem, definitely you can easily identify the disease and you can take remedial action. So this is a pathway to explore it or investigate the disease problem in a particular field. So management, how you will manage, as I mentioned earlier, sanitations. So because your stubbles or previously cultivated plant residues and weeds can carry the inoculum. Therefore, sanitation from the initial point of cultivation through the solarization and plowing and removal and burning of weeds and other residues highly minimize the future disease incident. This is sanitation. Second one, to sunlight, I told you, is the solarization. Third one is reducing seed rate. I told you the reducing seed rate mean, so if you are using the more seed rate, the plant will get dense. The plant will, if it is dense planting, there will be more chances for this disease and blast diseases, right? For any disease. So commonly any diseases and leaf hopper attack or viral diseases you can expect if the seed rate is higher than the recommended rate. Why? The hoppers can hide. So plant will grow higher for the competition of the light if the seed rate is high. Therefore, you have to maintain optimum density of the crop and drain the field. In the maximum tiller stage, you have to drain the field for a certain period, right? If you are drain the field, mean there is no chances of sclerotia to get in touch with the sheath. So that's how you can easily manage. So you observe, you are suspecting that your field or your paddy plant will get infection, then the draining the field for that the maximum tillering stage is an ideal idea to escape from this disease. And biological control agents. So if the field is rich with trichoderma and other fungal antagonists like pseudomonas and mycorrhiza, so they can able to minimize or mean that so your soil will become a suppressive soil. Therefore, the sclerotia will die, or the fungal mycelium or the bad fungi will removed automatically from the suppressive soil because of the antagonists, so antagonistic substances produced by this beneficial organism. So these are the way easily you can get. This is a common practice. Mainly, if you are maintaining these practices, easily you can get rid of from the many diseases. Right. So next disease is a sheath rot. The sheath rot and sheath blight is more or less same, but it is confusing. The causes are different, right? So the sheath rot is caused by the Serocladium, the Serocladium oryzae, right? Serocladium oryzae. So normally the panicles, right? Rot growing panicles are incompletely exert with an empty grains. That's the problem is the sheath is get rot. Like the sheath is get rot, so the disease will go inside and it will damages all these panicle and other areas. Therefore, the pollen get aborted. So then you cannot expect that your panicle is fertilize and produce grains. That is a problem. But this disease is usually associated with the virus diseases. The economical importance of these diseases is very less if you are taking in the Sri Lankan conditions. Right? This is a sheath rot. So, panicle initiation will not complete. If the sheath rot, so the panicle will not come out, look like the symptoms is. Management of this one, so you can use the resistant variety and systemic fungicide like carbon diazem or the mancozeb. It can work easily, and also you have to be treat, right? You can eradicate the seed borne. This is also the seed borne disease, so easily you can eradicate if you are dipping these seeds with the antagonist fungi, or even you can use for these fungicides, Captain and Therab too. Another disease is a stem rot. The stem rot is also caused by some of the sclerotia forming fungi, right? Sclerotia forming fungi. So normally the stem rot and the sheath rot, economically not important for while considering the Sri Lankan conditions, but the water zone rice, right? Stem rot is first appears at the tillering stage as a black lesions. You see, it's a black lesions. So in the tillering stage, and in the if your rice field is transplanted, right? If your field is transplanted, sometimes you can expect that disease, right? So this the disease is progressing, you are infected sheath and die, right? And slow off. If the sheath is died and the infection may penetrate in the culm, or insect may the infection may penetrate in the culm region. So if it is penetrated in the culm region, it means where it is attached with the seed. So this is progressing, so of this disease. This disease all causing fungi also producing sclerotia. So therefore, if you are taking any management aspect related to the seed blight control, easily you can control the stem rot diseases. For this disease also, the careful monitoring is highly appreciated, because if it is sclerotia formed, then management is very difficult. For this disease too, also you apply potassium fertilizers, right? Potassium fertilizers is highly recommended. So because potassium can induce the immunity to the plant. Normally the plant is in the poor soil drain conditions, right? Your soil is poor drain condition, definitely you can expect this sheath blight and the stem rot, right? Sheath blight and stem rot. So other sanitation procedures and fungicide application you can manage, but don't worry about in Sri Lanka because this disease incident is less. And the false cement. This false cement also you can expect here and there, even in the Sri Lanka, even the northern Sri Lanka too. But people thinking that if the field, rice field is showing that false cement, the farmer will get higher yield. The higher yield time only farm people are thinking that the false cement can occur. Right? Sometimes we call it as a rice fruit. Farmers will tell it's a rice fruit because this is a Bacterial mycota fungi causing this disease. Right? So normally their target is in endosperm, right? The target is endosperm. That's how this disease will, this endosperm will get burst, look like fruits, the bigger one. But from the bigger one, the endosperm, you can expect more amount of yellowish brown spores, right? So normally you see velvety spores enclose the floral part, right? Enclose floral part, the mass of yellowing fruiting bodies. That's how these spores become yellowish velvety, become yellowish velvety. And here you can see this incident. You could have seen that in rice field, right? If the rice, the farmers believes that this is they are not telling that this is not a disease. They are telling that this is because of the higher yield is happening because of higher yield. But actually, this is a fungus, cement fungi, the causing this disease, right? So if you are taking that, so normally the seed germination, so this fungi is causing the seed germination, poor seed germination, 35% of reduction. If your cement disease attacked paddy is harvested, and if you are thinking that you can use for the use as a seed paddy for the next generation, definitely you will not expect that you will not you can expect the germination loss 35%. And damp weather conditions, you can expect 25% of these disease. So in India, this disease yield loss is 70 to 7 to 75%, but loss is less in Sri Lanka, right? So favorable condition for the disease is high humidity, high nitrogen content, and wind-borne spore. This is not, it's an airborne disease. It's an airborne diseases. So presence of the fungi as is chlamydospore, not as sclerotia. Commonly you can find chlamydospores and uridospores and teleospores, because of the cement fungi, basidiomycota, it can produce more amount different kind of spores, right? The flowering stage, your field is will get infection. Your field will get infection at the flowering stage. So pathogen, normally Ustilla gonidia virens. We call it as an Ustilla gonida virens. This is an cement fungi, and Claviceps oryzae. So in the sexual stage is formed by the Claviceps oryzae. This is the size of these fruiting body and mycelium. So normally it produces elliptical conidia. The size of the conidia may be from the diameter 3 to 5 to 4 to 6 when to measure these pathogens forming conidia. This is a conidial structures produced by the smut fungi, right? Different conidia, different shape. This is a urodospores, right? This is a urodospores produced by the fungi. Number 5, this is a my single mycelia, mass of mycelia. It produces, you see the fruiting body, inside that you can find the spores, right? So this is the fungal color. So this is Citipite stomata, right? It's producing the Citipite stomata with globose head grows from the sclerotia. That's the thing. Here, Stipite stomata, through the Stipite stomata, so mycelium will grow. From this end of the mycelium, you can find the spores, right? Spores. So this is producing then ACMs, right? ACS spores also, but as not the ACS for ascaspores. But this is normally the cement fungi belongs to Basidiomycota. Sometimes it is confused with people are telling that it's an Ascomycota too, but now it is classified as an Basidiomycota. Don't worry about these things. This is a normal disease cycle, right? So normally from the chlamydospore, from the chlamydospore, if it is blown and if it is reached at the flowering stage, so that from this chlamydospore it will produce mycelium, and it can produce draw the nutrition, and infection go through the panicles, and flowers will get infected, and spore either infect developing spikelet or mature grain. If it is a mature grain is infected, definitely the endosperm will get burst. The endosperm will get burst, and cottony yellowish mycelium with conidia is like a big fruity nature, is an identification of this disease. So management principles. So normally you can use, right? Normally you can use sanitation, but incidences of this disease in Sri Lanka is very less. Therefore, you don't worry about that. But you can go for one shot. You can go for a one shot fungal spray if you are suspecting that adjacent field is having this disease, right? Otherwise, or at the time of panicle initiation or the flowering, not spray after on the client filling stage just started. Normally, you can go for the copper-based fungicides. The copper-based fungicide is highly recommended and not toxic to the human being than the other fungus, right? Not, or not. You cannot. You should not use the other fungus or mango sap because it's a poisonous one, because that attack is target is the flower. But if the disease incident is less, you need not to worry about that one, because immediately you will harvest. But if the disease incident occurred, it is better to avoid to use this harvest as a next generation seed. That is an idea. And also after the harvesting, you should burn the field. You should burn the field, so because otherwise you can expect the next season get infections. Another one is another important disease. This is also an important disease. We call it as a rice bacterial leaf blight, right? Xanthomonas orsicola, right? Xanthomonas orsicola. You see that the leaf bacterial leaf blight. So normally you can expect this one from the seedling stage, right? Until harvest you can expect. The flat leaf it's got infected, then definitely you will get then more yield loss. Right? So leaf blight, the symptoms like a water-soaked yellowish stripes on the leaf blades. So initial, this is a bacterial disease, not a fungal one. Initial disease incidence will start with an water shock streak, light yellowish streak, and definitely it will become a wider and lengther. Right? It will become a wider and lengther. There are 5 types of symptoms you can expect. The normal type, first as a dark green water shock, short infection threat. This means light yellowish marking. If you're observing the light yellowish marking, then you can think of that something is happening. From that you can take, oh, what you can do is, if this is a bacterial disease, difficult management is sometimes is difficult, right? Management sometimes difficult. So but you can go for even the trichoderma spray, because this is a fatty field, it's in the underwater condition. No, you can go for a trichoderma foliar spray or pseudomonas foliar spray at the initial stage. But if you are not taking care, it will advance, the whole leaf will become a dry off, right? Certain stage. This is a second acute type, right? So an acute type occurs environmental conditions are suitable on susceptible plant, susceptible variety, environmental conditions are suitable. So the disease leaves get dark green, rapid expansion within a few days, and gray or green. So earlier yellowish one, then again it is going for a dry off, right? Again go for a dry off. So bacteria, you can observe the bacteria as a yellow bead like bacteria honey. So right, oozing of the bacteria as a milky dewdrop from the lesion is an identification or the indication that this is bacterial disease. So this you can, from the symptom you can confirm this is bacterial leaf streak. The midrib, the wilting type. So wilting type is not general, but in the advanced cases plant can get wilt. The midrib type also slow symptoms, right? It shows symptoms in the midrib portion. It's also very less, but the leaf is highly get infected, right? Highly get infected. If the midvein is get infected, definitely the whole plant will get dry soon, because it will transfer through the xylem vessel. It will move to the all over the plant. If you are observing the symptoms, removal of the plants and burning is an ideal management option, because for the fungicides will not work. This because this is a bacteria, right? Bacteria. This is an because of this from this is a bacteria. No, if the plant leaf infected, the infected area will dry off. From that one, you can find saprophytic fungal growths. Sometimes farmers will confuse, then they will think that this is a fungus. They will start to spray fungicides. Therefore, the proper monitoring from the initial point is important to take care of the plant. Otherwise, many misconceptions will happen. You cannot manage your disease properly, right? Seedling wilt. If the seedling stage, seedling wilt you can observe, but in the observed after that, if your plant, if your field is get transplanted paddy, definitely you can start with the water shock and greenish mark, right? Greenish marks and dry yellowish color streaks and dry off. This is an indication. Yellow color streak, and the symptoms you can find the dewdrops. Then next 2 days, these areas will dry off. Therefore, the green matter content will get it off. Then the photosynthesis will less. Then it will lead to the severe yield losses. Right? So this is in the, if it is leaf, it's dry off, your panicle will be sterile and it will not form properly. So because this bacteria also causing the pollen abortion, one thing. Second thing is photosynthesis is less, then definitely you will get less yield, right? This is a symptoms. First symptom, second symptom, in advanced symptoms, right? First symptom. The problem is, if the bacterial leaf streak sometimes will get masked if other insect incidence is there. If, for example, leaf folder, Nephlochlosis medinalis, they are scrubbing and eating the leaves. If it is scrubbing and eating the leaves, act as an it's a given option for the bacteria to get penetrate. Right? Bacteria get penetrate. First thing. Second thing, the bacteria will get in through the hydrodotes, right? Hydrodotes or pores in the leaves. So bacteria need damages to get in, like not like a fungus. Therefore, this condition, particularly in the wet season, only you can expect this disease. Here, so the wilting in the seedling stage. This is you can expect, you will see the leaf is getting yellow. Sometimes this yellow confuse the farmers as a thrips incidence. Thrips incidence field is also look like this. But you have to be very careful. Thrip incidents mainly occur in the leaf tips, but this is you can expect the leaf blade, right? Leaf blade, but if it is symptom advanced, it will confuse with many diseases, right? Whether it is viral disease or the bacterial disease or the blast disease, right? So we have to be very careful, but early monitoring is highly accepted, right? So pathogen is a Xanthomonas oryza, Padova oryzae. So bacteria causing, so rodsep bacteria. I would have studied in the bacteria, right? So the rodsep bacteria. So these single pairs of bud chain. So bacteria can be there as a single cell, as a paired cell, as a chain. It's a gram-negative, non-sporing bacteria and develop a capsules. See here, Xanthomonas oryzae. You can find single polar flagella bacteria. You would have studied in the microbiology, right? So they can able to move through the seed or plant residues, even water and soil, right? So it can able to move. So I mean three mode of movement. So any movement, not an insect, to other mode of other mode, it can move and target the host, right? Target the host. So in the alternate host, these are the weeds, right? The Listeria and Zizania, these are the perennial weeds. So in these weeds also, you can not the perennial weeds. This is grasses, right? In these grasses, also you can expect this disease. Act as a harbor for even the off seasons. Right, so factors influencing the development of these diseases: presence of the wheat, presence of the rice stubbles and ratoon, right? Because bacteria can survive, no? And presence of the bacteria in the rice paddy and irrigation canal. So this bacteria, if it is there through the water and irrigation canal, it can move and your field get infection. And warm temperature and high humidity, like a fungal condition. The disease favorable for the fungal also. The bacteria can easily move and to the field and initiate the infections. Therefore, common commonly for if you are thinking a disease, the conditions normally except viral. For other diseases, warm, humid condition favor the bacteria and fungal and nematode disease. Right? The viral diseases it need dry seasons. So that's the thing. So you should always remember that for the hot, humid condition and humid condition favor the bacteria and fungal and nematode diseases, mainly bacteria and fungal diseases. Therefore, so the rule is, so you have to remove these weeds, right? Listeria and different species of Listeria and other diseases, other Cyperus and Cyperus diphormius, rotundatus, those weeds also you have to remove it. Right, so disease cycle, for example, if you are taking the disease cycle, so normally bacteria, so any source may be from the stubbles, so seeds may be seeds, disease seeds and water or anywhere, straw. So bacteria get through these sources, and if it is reach the plant leaves, right, may the water or it may be stubbles or the plant surfaces. If it is getting the plant surface, it will move into through the hydrothodes, so the wound created by the insect. That's what I told you. Nephlochrosis medinalis, right? Nephlochrosis leaf folder or disease or any wound created, it can go in high temperature and humid conditions. Bacteria will multiply very quickly. So if it is quickly multiplying, what will happen is bacteria plug the water conducting cells. Right, I told you it's going inside the xylem and blocking the xylem vessel. So if it is xylem vessel is blocked, then those areas will get built. So sometimes this part is blocked, it will dry off. Right, the part will dry off. Initial symptoms you can expect the leaf lesion. Then from the leaf lesion you can observe the bacterial zooes drops, right? Drops, and wind and rain favor the movement of the bacteria from one area to another area. This is a disease cycle of this bacterial leaf strip caused by Xanthomonas oryzae, Padova oryza. This is leaf streak. Another one is there, narrow leaf streak. This is caused by Xanthomonas oryzae, Padova oryzicola. Right? Padova oryzicola. This bacterial leaf streak is some wetland areas is high, but in dry zone is less. Right? But dry zone of Sri Lanka is less. 60% of the Asia's rice production, right, reduced because of bacterial leaves. Therefore, this is a very economically important disease. In Japan, right? In the Japan is normally you can expect this wetland rice cultivation. The environment is good, and also they are cultivating high yielding varieties. They are very susceptible. Therefore, so every year 3 lakhs to 4 lakhs hectare land is get infected by this bacteria, this leaf. So the overall disease loss due to this rice disease, which mean bacterial leaf streak, is 20 to 50%. The Indonesia is lower than the Japan. The Indonesia is getting lower than the Japan. So this because of this condition. So even Sri Lanka is very less than the other countries. Even India also getting infected, but in Sri Lanka, yield losses related to this less than 6%. Less than 6%. Right, so the problem is if the bacterial leaf streak is there, you will get less yield. You will get less yield because of blockage of the xylem vessels and drying of the leaves, no photosynthesis, blockage of the leaves, blockage of the transport of these food and nutrition. That's of the yield loss is very high. Yes. So those are the things we have to be very careful while observing these disease. The nitrogen conditions, so nitrogen and resistancy of the plant, and ooze conditions whether there is there, and the leaf dry of water shock mark. So all these observations are very important. So early monitoring with these observations will indicate or give an idea that what is the cause. So management aspects. So because this is a bacteria, management is very difficult. So therefore, you have to go for an early sanitation and removal of volunteer plants. Early sanitation with a volunteer plant, and so if the bacterial disease observed, you should cut the water movement from one area to another area and fully dry off. Right, fully dry off and minimize the insect incidences and optimal level of balanced fertilizer application. Balance. You should not apply nitrogen. If it is a nitrogen, it will provoke the disease. The use of resistant varieties is highly important because they are trying to develop bacterial leaf stick varieties in Sri Lanka too. If you are cultivating resistant varieties, you don't worry about that, but you need not to apply. But the people they are using in India, right, bleaching powder, zinc sulfate, but it will not work. Right? It will not work because this is a bacteria. But if you are giving the zinc sulfate and silicon, sometimes it will increase the immunity of the plant to minimize the disease. So what you can do is pull out the disease, drain and saturate the soil, apply minimal amount of nitrogen fertilizers. These are the things you have to take if you are observing the symptoms in your paddy field. Right. So next things you have to be careful. Neighboring field is there, they then block the water. Right. If the neighboring field get infected, observe the symptoms, block the water to enter in your field. Right. Block the water, enter your field, and drain off the water present in your field. Block the coming and drain off all the water. And so you should maintain after the harvesting with a fallow period, right? You have to maintain harvest fallow period with a very dry to kill the bacterial cells in the stubbles, and stubbles and residues should burn properly, right? Burn properly. So this is the second part of the disease. So we have to take another one more classes to complete these diseases. So I think you would have learned many important diseases in these two classes. So important diseases, once I am remembering that the rice blast, the brown spot, and sheath rot, and bacterial leaf streak. These four diseases are economically important even in Sri Lanka. So therefore, you have to be uh, replenish and fill your knowledge while repeated reading of these notes regarding these economically important diseases. If you this is an end of the part 2. If you won't ask any questions, please ask.